Thank you very much. So yes, I'll be talking about not just topological data analysis, but how it's really integrating with statistics. And so I'll start with a brief background for some of you, probably a bit of review on what topological data analysis is and some persistent homology, but then I'll move into persistence and statistics, so how they've integrated, and then I'll talk about topological modeling of surfaces in particular, and then I'll talk about kind of where TDA and statistics are going in the future. So I always like to start with this slide to get everyone in the right mindset of topology. So the common joke is that topologists can't tell the difference between a coffee cup and a donut. And that's because, as you can see here through this continuation, that we have, we start with the coffee mug, work our way around with a continuous map, and we maintain that one particular hole, but otherwise everything kind of morphs into the donut, and it can continuously morph back. So the idea of topology being integrated with data analysis is we want to capture the big picture of what's going on in the data and not get too caught up in the noise of the details. Now, one thing I'd like to point out is there is a curvature to the coffee mug that allows you to hold coffee, whereas the donut does not have that kind of curvature. And so one of the things I'll be talking about later is how we integrate both topology and geometry to fully capture the data, because sometimes topology can miss important things. So just a general overview of what TDA is. The idea is that we're building these new data methods that is grounded in rigorous mathematical theory, but still gives us new insights to our data that statistics alone cannot give us. And for those of you who are biologists, stay with me. We're going to have some math first. But I will be showing examples of how we can apply TDA to viral evolution, protein conformations, histology images, and finally, heel bones in primates. So any TDA algorithm you see is going to follow the same basic outline. You're first going to have a finite set of data points that is in some sort of metric space, or it just has a notion of similarity. And this gives us a bit more flexibility than statistics because we don't necessarily have to be in a metric space to start or have a sense of the geodesic that's underlying the metric space, or not the metric space, the data. So then we build a continuous shape on top of the data to highlight whatever the topology or geometry is underlying underneath. Continuous is in quotes just because you sometimes get shapes that are disconnected, so therefore they wouldn't be continuous. But in general, that's what we're building on top of the shape. And then we extract the topological and geometric information. The important part of this step in a TDA algorithm is proving that this part is stable to noise or outliers. So that's a key component you will see in any TDA paper when they're presenting a new method. And then finally, these extracted features can give new families of features, and this will hopefully give us insights that is, well, one, captures the information that statistics did, but also could give us a new angle that we couldn't get before. So some basic topology terms that you'll need to know or probably have heard before, but we'll just review. So up top here, we have the building blocks for a simplicial complex. So we have our zero simplex, that's just a vertex. That one simplex is just an edge. And then we have the two simplexes is a triangle, and then our tetrahedron is the three simplex. There are 4 and 5 and 6 simplices, but we don't normally picture those because those are in dimensions beyond 3D. So a simplicial complex is just anything that can be built out of these basic building blocks. And then you're going to hear me talk about homology groups and Betty numbers today. There is a rich algebraic structure underneath this that I won't go into. But what you need to know is the homology groups capture different aspects of our shapes. So the zero homology captures the number of connected components. The one homology group will capture the number of holes we have. And then the two homology group will capture the number of voids we have, so like the hollow inside of a sphere. These are the features that are going to be preserved under homeomorphism. So these are like what I was saying before, where we want to capture big picture ideas without getting too caught up in things like the size or the exact border outline of a shape. Finally, the Betty number, you're going to hear me using that word a lot. That's basically, in math speak, that's the number of generators for the homology group. In application, that's just the number of each topological feature that we can count in our shape. So zero homology, the Betty number here is 2 because we have exactly 2 connected components. Beta 1 here is 1, sorry. The first Betty number is 1 because we have 1 hole, and then we also have 1 connected component, so Betty 0, 1. And then finally, we have beta 0 is equal to 1 here because one connected component. We don't have any holes, but we do have one void, so that is beta 2 equals 1. Are there any questions so far, or do we have everything? Beautiful. Okay, so when we're doing our persistent homology, we start with a point cloud of data. Points in space, that is our C. In our barcode, you see that we have a single bar for every single one of these points. And this is for the 0 homology. Every point starts out as its own connected component. Now we're going to run a parameter epsilon and start building balls around every single point. And you can see as that epsilon parameter gets bigger, some of our connected components start to merge. And this is reflected in the barcode diagram. These features essentially die, whereas others persist on. Now, as we continue this filtration, we start to see 2 loops emerge, or holes, so that's where these blue bars are coming in. And then finally, as we go along, we see that we just end up with one giant connected component with no holes or voids or anything like that. So that's usually where we stop our filtration because there's no new information to gain. Part of the reason we run the parameter all the way through the filtration is so that we don't have to have a user-defined epsilon before extracting the topological information. So that gives us a fuller picture of the topology, but we also don't have to rely on users to pick a parameter and say, oh, that's where it's different. And finally, this persistence barcode is usually stored in a computer as a persistence diagram, which is just a list of birth and death coordinates. So each of these points corresponds to one of these bars, and the x-axis is the epsilon parameter where the feature was born, and the y-axis is the epsilon parameter where the feature dies. You'll notice that all of these lie above this x equals y diagonal. That's because you have to be born in order to die. So that captures the full persistence diagram and the entire filtration. This last feature here is almost always that just giant connected component we have at the end. Any questions on that? Beautiful. So to give you guys an example of a filtration, World Series just ended. So we have 2 different baseball defenses. So normally a baseball defense will line up in these positions. But if you have a player like David Ortiz step up to the plate where he hits 2/3 of his hits to right field, you might have a shift here. And so if we want to say, well, is there really a difference between these 2 defenses in terms of the geometric or topological structure? What we can do is we can start this filtration with balls, and you can see as some of the features are starting to emerge, you got some edges, now we're seeing faces. And so the idea would be is to derive the persistence diagrams from those features and then actually compare them. And so our means of comparing is either the P-Wasserstein distance or the bottleneck distance. And the idea behind these 2 distances is that you have to pair off all the points of a persistence diagram, and you're looking at how far you have to move each point to get to the other diagram. So the idea here is that we'll say, okay, well, is it closer if I pair these 2 and these 2, or if I pair this with that one and this with that one? You're taking the infimum over total movement overall. Most people in the literature use the bottleneck distance because it is far more computationally efficient than the P-Wasserstein distance. But that's our means of comparing them. You would probably run a hypothesis t-test to see if there's like some sort of significance in those distances. So you would try to switch up points, get different diagrams, and then compare those distances. And that's how you would say, okay, there is a significant difference between these 2 persistent diagrams. So to give you guys an application, we'll talk about viral evolution. So the standard way to represent evolution in general has been the phylogenetic tree that dates back to Darwin. But with viruses, we can have horizontal or reticulate evolution. And that's tough to capture in a phylogenetic tree. I mean, yes, you can see here that we have 2 branches that come together to make a new virus, but that's kind of misleading because viruses don't mate or reproduce. So we want to say, is there some better way to capture these kinds of complex horizontal events? And so the idea that the authors of this paper had is that each genetic code you visualize as a point in space. They did this with a technique called principal coordinate analysis. And then they used a genetic distance as their parameter. And then their goal is to hopefully capture the complex exchanges with more than 2 organisms or viruses. And the way they interpreted their data was 0 homology was your vertical evolution, so that's your traditional tree. Your horizontal evolution was the one homology, because you can see in the tree you start to get holes. But then when you had complex reticulate evolution, where 3 or more came together, that was their two homology group.
So
yes?
When you're talking about holes here, you kind of ignore the directionality?
Yes, usually.
Yeah.
So directionality is geometry, not topology. So great question, though. So in this simulation, they actually, so the top one, they did only vertical evolution. Yes?
Say more about why the complex event corresponds to void.
So they say they said that corresponds to a void in part because it was kind of ended up being multi-hole here. If you look at the tree where 3 different viruses would come together, then you've got 2 holes here. And so you could capture it with 2 holes, but they thought topologically there might be more going on under the surface. It's more complex than what one homology can capture. And so that's why they went up to that higher dimension to better capture the complex evolution.
Did they observe persistent homology in the data?
They did. I will get to that. Great question, though. Any others? Cool. So this is a simulation data. So they just did vertical evolution for the top one, and they ended up only really getting connected components. And then in the second row here, we see that they added horizontal evolution, but just of 2 components or 2 viruses coming together. And that's where you start to get these holes in your filtrations of the data. Now, when you move to actual data, such as avian influenza, now we're starting to finally capture that dimension 2 complex reticulate evolution. And that basically comes out, we can have now a geometric or topological representation that more accurately captures how the virus has evolved. So here we see it as a network, so it's not necessarily a tree because we do have all that interconnectedness. And then in the bottom one, we have a 2 simplex that kind of shows how things evolved out and then came back together. So if there are any questions or, beautiful. So with comparing persistence diagrams, there is a bit of a downside in that it is computationally expensive. Even for the bottleneck distance, which is much better, it's not great. And that's because we have to take the infimum over all possible bijective maps between 2 persistence diagrams. So when you only have 2 points on each persistence diagram, that's not bad. But when you have actual data, you're going to start to get 40, 50 different pairings, and then you have to check every single possible pairing among those points. And so that ends up building up pretty fast. And in fact, your computational efficiency ends up being m to 5 halves times log m, where m's the number of points in a persistence diagram. So if that's the case for every single persistence diagram pair, then you can see where it computationally adds up if you're trying to cross-check 40 persistence diagrams against each other, or 100 persistence diagrams. That distance matrix becomes very tough to calculate. Doable, but we can improve that. In addition, the space of persistence diagrams doesn't necessarily let us have a unique mean between a set of persistence diagrams. So these 2 before, the way you would calculate a mean is you do what's called a Fréchet mean. So again, you would pair them off and say, okay, what's the middle ground between every pair of points? The problem is you now have 2 equally legitimate mean persistence diagrams. And this makes it really tough to implement statistical methods, to put probability measures on the space. And in practice, it makes it tough to integrate with those statistical and machine learning methods. But we don't want to give up because, like I said with the viral evolution, we do gain information from the topological data analysis. So how do we move on from here? So like I said, we want to keep going because we can look at an underlying manifold. It gives us new information that can't be captured necessarily by traditional methods. We do have a descriptor in a metric space. Those 2 distances from before are true distances. So you have the only equals 0 if they're the exact same, you have symmetry, and then you have the triangle inequality as well, which are very beneficial, at least when we're trying to compare similarity between 2 objects. And finally, it is stable against outliers. If you have points that are far off from the other ones, you'll end up just having extra connected components. They won't necessarily form holes or anything like that, so it won't interfere too much with your data. However, like I said, It's tough to integrate with the statistics and machine learning tools we have. That's a result of having a metric that's, one, hard to calculate, and two, we don't have a guarantee of a unique mean in a data set of persistence diagrams. But we kept up with it, and now persistence landscapes came along in 2015. It's the same start as before. We have our set of points, and we run our filtration till we eventually have one giant blob. And so if we look at this persistence diagram, only the first homology group is shown here for simplicity. We then drop a line from every single point to the diagonal, and then a point across to the diagonal as well. And what this does is it ends up partitioning the region of the persistence diagram by the Betty numbers. So this region and the region below the diagonal are where the Betty number is 0. There's no holes, there's nothing really interesting. But then we have Betty number 1 in this region and Betty number 1 in this region that corresponds to these holes separately. Where there's this hole alive and then there's just this hole alive. When they overlap, that's when we have 2 holes. And then there's this brief overlap here where we have 3 holes temporarily, and so that little region is captured there. All right, so this next part, there is math that underlies it, I promise. We're gonna tip over the diagonal and fit it to the x-axis. And so what this does is we have each landscape feature starts to emerge at its birth time and then finally ends at its death time. And this is along parameter epsilon. The y-axis doesn't ever have a label in the literature. That's just because it's a scalar that's proportional to the lifetime of each feature. And you can see here, I put the barcode here as well, so that you can see that if you wanted to derive it directly from the barcode diagram, you can. So you see, like, for example, this 3 over here matches when there's 3 barcodes overlapping here. So now that we have this, we can then label every single one of these functions based on the Betty number region it bounds. So for example, this lambda 1 bounds the region where we have Betty number greater than or equal to 1. And then this red function here bounds that region where we have Betty number of 1 greater than or equal to 2, and so on. There are lambda 4, lambda 5, lambda 6 functions. In this scenario, they're equal to 0 because we never get Betty number greater than 3. And then you have 2 choices for representing this. One way is to do with the flat functions like this, just all overlapping. But a lot of people choose to use this 3D representation so that you can actually see everything build out. And then you also get that visual of what the Betty numbers are doing as well along that third axis. And then we have a distance now. This is just the p-landscape distance. You'll see here that it's a lot of ugly math, but it's the same math as before, just no infimums, no bijections, which makes it computationally a lot easier. In fact, we're now at OM squared for our distance, where m's the number of persistence points in our diagram. One thing to note about this is that it is actually a lower bound for both the p-landscape and the bottleneck distance. So we do have a way to relate back these persistence landscapes to their original persistence diagrams. Again, while saving that calculation time. So another advantage of persistence landscape, oh yes. Yeah, so this is basically an LP distance between 2 functions. Yeah, so you're summing over every single difference function. So like you would sum the lambda 1 distance, then the lambda 2 distance, et cetera, over LP. So good question, thank you. So anyway, so I was saying we have unique means with persistence landscapes. So if we take the 2 diagrams from before, which did not have a unique mean, and we calculate their 2 respective persistence landscapes, we do get a unique mean here. And so now we can actually unlock a lot of those statistical tools that we want to use, including hypothesis testing. You'll notice that there's kind of a plateau on this persistence landscape, and that this doesn't really correspond back to a persistence diagram. That's similar to how a Poisson distribution might have a mean of like 2.7 or 1.5, even though you're sampling on the positive integers. So that's a lot. All right, so now that we have a unique mean, we can do this, we can apply this linear transformation to it, and we now have a random variable that we can work with. So persistent landscapes are in a separable Banach space. For those of you who are not mathematicians, that just means that we can apply the strong law of large numbers and our central limit theorem. And that means that if we have enough samples, we can assume that we have a Gaussian distribution when we are studying those persistence landscapes. And then in particular, when p equals 2, the space is also Hilbert, which means that we can now have a positive definite kernel, and that allows us to apply kernel machine learning methods. So now we've unlocked the statistics side of things, which is great. So the example I'm using for this is conformations of the maltose binding protein in E. coli. So a protein can have either an open or a closed conformation. That closed conformation tends to be much more stable than the open conformation, which means if there is ligand binding, it's more likely to hold onto it and transport it properly to its destination. And so one protein can be represented as 370 points in R3, and so that ends up being the input for a dynamic cross-correlation matrix, and that's what you end up inputting into the persistence homology filtration. So again, they used it for 7 closed and 7 open conformations of proteins. They weren't able to do it for many more because that data is very expensive, so they actually had to use a 2-sample permutation t-test rather than just assuming it's a Gaussian distribution. And their null hypothesis was that, okay, the mean persistence landscape for the closed proteins conformations is going to be the same as that of the open conformations, and the alternative hypothesis was that it was not equal. And finally, they also classified using support vector machines. So again, back to that statistical use that we have. They used 50 points from the persistence landscape. So along that parameter axis, they chose 50 different points, and then those coordinates formed that vector where they trained their algorithm. So just to give you a visual of these proteins, so here on the left, you can see the closed conformal structure. There's that ligand right there I was talking about, whereas over here you have that open structure. And the yellow indicates the active sites of the protein, which are involved in holding onto the ligand. So these are the resulting persistence landscapes. Up top, we have the zero homology group, the connected components, and the closed and open are closed on the left, open on the right. The bottom row is the first homology group. You'll notice the general shape of the landscape are pretty similar. It's really when you get down to these higher Betty number regions where you start to see the difference. And it was picked up in a hypothesis testing. In addition, they were able to correctly sort all 7 closed from all 7 open conformations. And I know what you're thinking. Okay, this is all statistics. What am I gaining from doing the persistence landscape? They actually found that these most persisting landscapes correspond to the region, in the first homology group specifically, corresponded to a ring that encompassed almost 80% of the active sites in the protein. So we did learn something about the structure of these protein conformations in addition to having these statistical values. Any questions? Beautiful. All right, so to recap persistence landscapes, we can treat them as random variables. We could not do that with persistence diagrams. That's awesome. Our distance is computationally more efficient to calculate, and it gives a lower bound for that P-Wasserstein distance and bottleneck distance. So that allows us to relate it back to the original persistence diagrams. And then finally, we can apply hypothesis testing and some machine learning methods. However, that vector form takes a little bit of extra processing, and it can also depend a little bit on how many points you choose. So you possibly risk maybe losing some topological information in the process. And in addition, it's a bit limited in what machine learning methods can be used. So we want to improve this. Which brings us to persistence images. So this is a visual of the process of how we calculate a persistence image in the first place. We start with our data. We start with our persistence diagram. You know, exact same start as before with the filtration and the calculations and the birth depth coordinates. We're then going to run a transformation, so we're going to make this scaled diagram. So this x-axis is still going to be birth, but this y-axis is now going to be persistence. And now we have a better idea of not just which coordinates are where and where they're born, but also the length of their persistence. So from there, we have to choose a weighting function, which will depend on the application. Topologists would tell you that you want to weight the functions that are most high in persistence. Those are the features that are most important and most dominant in the data. However, in experiments, such as in neuroscience, they found that it's the middle persistence points where you find the most difference. So if you know something about your data, if you know which parts of the persistence diagram you want to look at, this is a great option because now you can actually weight points, which you couldn't do before. The next step is to actually choose a probability function over R2. Most people use a joint Gaussian with different distances of sigma, or different parameter values of sigma squared. It's been found that the sigma squared choice doesn't usually make a difference, but it's good to check at multiple values. And then the mean is usually a sum over every single point in the diagram. So it's almost a mixed Gaussian type thing. And then use both those to calculate your persistence surface. That's what's visible here. So what you're doing is you're basically taking this persistence diagram and saying, okay, now how important are these points? And you're making a continuous surface out of it. And from there, you divide that surface into a grid. This can be as coarse or fine as you want, depending on the application. But now, the persistence image is the collection of pixels that you get by integrating that surface function over every single pixel. And so I mentioned before in the persistence landscape that you might lose topological information when you go to vectorize. Here, we've put all of the topological information directly into vector form, which means it's now good for any machine learning methods, not just kernel-based methods. We can use stuff like random forests and decision trees.
Right.
Yes?
So I was thinking about 4 slides ago, so I did not quite get it. Why are, well, here's the stupidest way to put it. In the first colored image under 2-4, why are some things like brighter than others? Why do some of those correspond to probabilities?
So these are higher values. So these are color-coded by like a gradation. So like this dark blue is probably a 0 value, and then it's builds up. It's like a heat map almost.
Yes, but those points get brighter colors because they persist longer?
So if you chose a weighting function that favors persisting features, then yes, they get brighter colors because they're more important.
And is that what's done here specifically?
Yes.
Okay, I see. Okay, cool. And then why on the right we're pixelating to different degrees of coarseness?
Correct, yeah. So up here is like the finest pixelation, whereas over here is kind of the coarser pixelations, where you get more of a grid.
Are we using multiple versions there, or that's just your...
This is just to demonstrate options in terms of coarseness and fineness. A lot of papers you see with persistence images do kind of experiment with that as well to see what best captures it. You're trying to balance, obviously with coarser pixelation, you get shorter vectors, so that saves you computational time. And so it is a balance between how much computational efficiency versus how much information do you capture. So that's kind of what's supposed to be captured in that.
Okay, cool, got it. Now, I'm going to ask my question from 4 slides ago that I was thinking about. I don't know the actual number of slides.
Yeah.
When we're comparing landscapes, yes, exactly,
yeah.
Perfect. So especially here, you're comparing features, that are born and die under the 2 different things. In this case, poles, and your 370 things are 370 axes.
Yes.
So do they look at all, here you're not asking what the features are, but you are somehow matching them up.
You can, so when you look at the persistence landscapes alone, you can't probably say, oh, this mountain is from that ring. But if you go back and watch the filtration, you can figure out where it's coming from.
Right, so do you know if here they actually look to see if the features that are lining up actually correspond?
They did, yes. And that's where they found that extra information of the active sites forming a kind of ring.
Okay, cool. And then also they compare the different conformations within closed or within open?
Yeah, so that's part of how I think it was the permutation t-test, that's part of how they figured out significant distances.
I see, but somehow they don't have enough conformations to compare within closed or something like that.
Well, they had 7 closed conformations and 7 open, so they could, but then they had to do a few more permutations,
Right.
so.
Okay, cool. Thank you.
Yeah. Any other questions? Yes.
I just want to make sure I understood correctly about before recording. So you're taking the picture in the middle, and you are fitting Gaussian mixture model on top of the picture.
Yes, with a weighting function.
With the weighting function, and then you are kind of doing some local average to get at some scale to get the pictures on the right.
Yeah, so you take every single you take every single grid square and you integrate over that, and whatever that value is what that pixel value is. So, yeah. All right. So the application for persistence images I'm going to show you guys is the histology image. So this is for colon cancer and cancer of the colon. And so the goal is to characterize kind of the glandular architecture of some of the histology images they had. And they used images from this data set, which was part of a contest. It was 165 images total, 85 training, 80 test. And what they did was they went through by hand and marked all the nucleoids in this image. And they used that as their point cloud data for their filtrations. For their weighting function, they use this function, which essentially is what topologists use. It favors the features which persist the longest. And they also use the Gaussian probability distribution, which is standard, like I said. So their official persistent service calculation was this lovely formula right here. Yes?
Sorry, so the point cloud is, what exactly the 7?
They marked the nucleoids within the cell images.
But it's what, an XY position or a list of XY positions or what?
Essentially, yes, a list of XY positions. So you can consider this in R2.
Okay.
So, yes, good question. So, yeah, and on the right here, I hope you guys can see the colors, but these green crosses mark all the nucleoids, and then the pink is just the histology image itself. And so these are the corresponding persistence diagrams, and then their persistence images. Someone asked earlier about different grid sizes. This one was 30 by 30, and I think this one was 30 by 40. So you had 900 pixels here, whereas you had 1,200 pixels in this second persistence image. One thing that they did not take advantage of, so they just used classification methods, and I think when they applied support vector machines to the histology images and their persistence images, they had about 85% accuracy, which was competitive with the contest winners. However, they really didn't take advantage of that new topological information. So as you can see with the healthy image, you have these persisting features that you don't have in this image over here. And so again, they're trying to describe the glandular architecture. This would be a great point to use if you're trying to do that in addition to classification. So persistence images are great. They take just as much computational power as the persistence landscape, including their distance, which once you have a vector, you just use those standard vector distances you have. But they're outperform persistence landscapes in classification tasks. And once you calculate, you have a vector, so it's pretty much applicable to any machine learning task you want. As I said, we have the computational efficiency and the distance calculations. And this, unlike the first two persistence objects I showed you guys, these can be tailored in parameters based on your application. So if you do know something about your data, if you know which features are important and which aren't, then you can actually get some better images and comparisons. So the problem is with the persistence diagram, it's hard to recover that from the persistence image. If we go back, this isn't really, I could be like, oh, I know exactly where all these points were plotted. Whereas you could do that with the persistence landscape. In addition, that persistence image distance can't be related back to the original bottleneck distance the way that the p-landscape distance can. So that's a con in that if you want to have the persistence diagrams, you're going to have to use that extra storage in your computer and keep that in the first place. And then in addition, we can improve computational efficiency for pre-processing into vector form. And this is an ongoing kind of field of TDA. It's called featurization of persistence diagrams. But essentially, it's how can we keep all the topological information, but get it into vector form as fast as possible. So now I'm going to switch gears a little bit. We're going to talk about topological modeling of surfaces. And I'm going to start with an example just to kind of give an idea of why we would do this. But if you have, let's say, a heel bone from a primate, if you give this to a topologist and say, how are these heel bones different? They might say, well, they are all homeomorphic to the sphere, and therefore they're the same, and we can all go home. But any evolutionary biologist would tell you that's not true. There's differences. So what we want to do, this is kind of what I was talking about before with the coffee mug, where we want to actually capture both geometric and topological information. And so that's where the persistent homology transform comes in. So we're going to start with a shape in Euclidean space. As long as it can be written as a simplicial complex, that's finite. It can be any shape we want. And then we start with a vector on the unit sphere. This vector is going to give us a direction to go in, and we're going to take a number of these unit vectors. And then we're going to run our filtration by almost scanning the image along that unit vector. And as we go through, that's going to be our filtration. We're going to be looking at how connected components emerge from different slices of our 3D object, and how, do holes emerge, do we have voids, etc. And that's how we're going to get our ultimate filtration. So you take these filtrations from however many number of directions, could be 10, could be 50, could be 100, and that's going to capture the full picture of the image. And so in this original paper, they used primate heel bones. So to illustrate, so like I said, we have this bone here. We have our first direction. We're just going to run our function across, and that's going to give us one persistence diagram. And then from a second angle, we're going to, again, run it across. That's going to give us a second persistence diagram. So we're going to build up these persistence diagrams, and that total data set is going to fully describe that shape. And by getting it from different angles, we start to capture some of the geometric information as well. And so in this paper, they actually used 67 different heel bones, and they were able to use clustering to recover the phylogenetic groups for these primates, just by scanning across heel bones and stuff like that, which is pretty cool. Another groundbreaking part of this is that this was shape statistics without using landmark-based data. So in previous methods, what you had to do is you had to pick points on your shape and say, okay, these are the features I want to capture. Now, for obvious reasons, this can lead to bias, and it can also lead to, oh, if I move this point, now it's significant, nailed it. But obviously, we want to do as little of that as possible. We want statistically significant result without user input, if we can. And that's what they were able to capture here with the clustering. Yes?
So can you explain again, this seems to be a key point, what is the difference in the persistence diagram when I pick one random direction versus another? I don't understand how the direction...
Yeah, so let's say we just have the shape here, right? So if I'm filtering across in this direction, if we just assume connected components, or even holes. So like here we have 4 connected components at this point versus 2 here versus 2 here. And at some point you have 4 as well. But then when we go from a different direction, there we go. We have 2 points all the way across the entire time. So that would give us some indication that there's extra curvature on this part, in this unit direction, versus from this side, there's no necessarily indents. Because if there were, you'd end up getting 4 connected components like you see here.
But still, I don't quite get the blob there. That's not my data.
This is a shape in 2 dimensions. So your data is just what the shape objects are. And so you're taking every single shape object and running this filtration in different directions. And then once you have that family of persistence diagrams for every single individual shape, that's when you can compare them. Does that make sense?
No, but I can figure it out later. I mean, it probably makes sense. I don't get it. I don't get how you can tell if you have just a connected component, for example. For my original data, I have data in RD, now I pick a random direction. How do I decide if I have a connected component or not? How does that relate to the direction?
What this slice ends up doing, so you're going to pick multiple directions, not just the one.
Sure.
So what I'm trying to illustrate here is that you end up having in your filtration these 4 components. That's where your connected components would come in, if we assume that this inside is empty. And then, you know, once we keep going in our filtration, then we end up having only 2. So you only have a... So that's when we transition from 4 connected components to 2. So therefore, these 2 have died.
This is like, when I'm doing this, this is as I go farther out along that particular direction?
Correct.
Fine.
Yes.
Oh, very good. Okay.
So, yes.
Are there any standard ways for setting the resolution of these grids?
So there is the need to make sure you orient your shapes correctly so that you do get the same directions for every single one. That has to be done by the user. So, and then in terms of the number of directions you choose, it depends on the application, but again, you're trading off computational efficiency versus how much information you capture. So, yes?
How's your shape given as a grid?
So you build a simplicial complex on top of it. So if I, again, had this blob shape, what I would do is say, okay, if I put points, so you'd say, okay, I'm going to construct it out of edges. And then, you know, if it's a surface like the above, you would end up using triangulation, so you'd get the 2 simplices. Yes?
How stable are the diagrams based on the filtering direction?
I don't quite know the stability of it. In general, persistence is pretty stable in general, especially to the small shapes. I would assume that it'd be more sensitive the less directions you use. So if you use more directions, it gets more stable. Yes?
Is there any or anything that shows that this could be, or the scale of analysis could be reduced to noise in a diagram? If you have a diagram in data,
Mm-hmm.
that may be a persistent analysis that could go on for.
Yes, so in terms of point cloud data, there are proofs for showing stability with respect to those distances. It does come down to a little bit how you project those points into space and what distance metric you choose to use. And a lot of the literature studies that in the pure mathematical TDA. In terms of the shape, at infinity, if you could use every single direction, this persistence homology transform is injective. So that means that if you were ideally in a perfect world going to get every single angle, then you would end up getting exactly 1 homology transform for that shape. Now, obviously in real practice, we can't use that, but we can get very close. And so even when you do have kind of weird dents or something, maybe you dug up an archaeology bone and you found a dent in it, that shouldn't make too much of a difference because the rest of the angles would cover for that. So, good question though. All right, awesome. So this brings us to the final part of the talk, and I'm going to tell you a bit about where topological data analysis in general is going. So in terms of the math side of things, there are people building up the theoretical frameworks that we can use more tools in application. So there's a group out of Michigan State looking at compactness of persistence diagram. That's a big topological characteristic, but it allows for other statistical and real analysis tools to be used. There's also groups who are looking at cohomology, which is a dual functor to homology groups, and looking at how we can use that to calculate persistence diagrams and whether that reveals any new information or maybe in some cases can get us to the persistence diagram faster. In addition, so not every single TDA application is persistence. There is a plethora of topology beyond just persistent homology. An example of this is network topology. So people want to know whether it's social networks or crystalline structures in glass, et cetera, looking at the topological structure of this. And so there's kind of more graph theory centered in that, but we're also capturing topology with that as well. And then there's a field of topology called knot theory, and that's very applicable to DNA and protein folding. A lot of rich stuff there. If you want to learn more about the math side of things and what's happening, there is the Applied Algebraic Topology Research Network. They run a seminar every other Wednesday online at 2 p.m., except when they have someone, say, from Australia, like Kate Turner, then it's at 5 p.m. But Normally they're online, they're really good talks. And then in terms of statistics and machine learning, there's also a lot going on there as well. This group out at Montana State, they're trying to build algorithms to learn the simplicial complex from the persistence diagram. So if we have that persistence diagram, can we go backwards? This group is looking at time series and dynamic data and figuring out how to apply persistence tools to that. And then also look at algorithmic integrity. So they're looking at, are these models actually accurate compared to real world data? There's ways to use topological tools to check your model. And then finally, there's groups in statistics who are saying, okay, what are some sufficient summary statistics for persistence diagrams? Because if we can calculate those, then we might not have to go through all the trouble of calculating the persistence diagram in the first place, which would save computational power. All right, so thank you to everyone who came today. Shout out to my advisor, Dr. Lorin Crawford, and I also want to shout out Alan, who's sat through, I think, 3 of these presentations and gave me a lot of feedback, as well as the NSF for funding my degree, and thank you all for coming.
So Emily, I think, did a great job in kind of like setting me up, and hopefully I do a good job in following through for the rest of this. I'm going to take a more statistician's point of view on this stuff. I'm technically trained as a statistician, but a lot of the stuff in my lab, we think about stuff from genetic and genomic variation as it applies to different traits and architectures of traits and phenotypes. So we do this a lot of times from a stat gen perspective, so like GWAS and those kind of perspective. But a big theme in my group is the idea of like dissecting variation of traits. And so typically we do this along the genome with some kind of genetic type measurements. In this talk, we're going to do the same thing, but the analogy here is we're going to do this with shapes. Okay, so I'm interested in studying how variations of shapes differ across different groups, and then relating these back to basic genomic and genetic properties or phenotypic properties that we kind of understand through biological principles. And so a lot of stuff that came up in the last talk about how do you reconstruct shapes, how do you kind of study shapes and do this idea of like variable selection with shapes. That's the stuff we're going to talk about today. And so any ideas people have here would be awesome, because this is kind of like the method I'm going to present today is like the first of its kind. And so any kind of ideas to like make it better or things that you think could be useful as a use case in terms of applications would be awesome. And so my presentation outline is going to be really building off of stuff that Emily just talked about. The first idea we're going to start with this idea of the history of comparing shapes in statistics. Emily briefly touched on this idea of topological summary statistics. This is a big place where I got my training in TDA, and I'll talk about this idea of representing shapes based on their topology using a different type of invariant called the Euler characteristic transform. I'll talk about a prediction-driven application that I did with a group at Columbia, Raul Rabadon's lab in radiogenomics. And then I'll introduce this idea, this new pipeline that we kind of came up with, Sinatra, and I'll explain where that name came from also, for variable selection with 3D shapes, okay? I'll talk about this algorithmic overview. Sinatra takes advantage, it's just a pipeline, and so I'm gonna detail my idea of like the steps in this pipeline. And keeping in mind it is a pipeline, so if you don't like any part of it, take it out and put your own favorite place in, right? I'm going to talk about this idea of rate, or this idea of relative centrality. So Sinatra uses what we call non-parametric or machine learning type methods to understand the patterns of, or the variance of 2 different groups of shapes. And then it uses this nice variable selection measure called rate, and I'll explain what that is. The cool thing about Sinatra is once you've transformed a shape into a topological summary statistic, you can actually, after you do some like variable selection in this topological summary statistic space, Sinatra allows you to actually map these significant measures back onto the physical shape itself. So it kind of reconstructs the shape, right? So you can actually do like variable selection. So if I was a biologist and I gave you like images or some kind of bones, it actually highlights the areas of the bones that best describe the variation between 2 groups. And then I'll show some simulations of how this works and some real data classification on actually primate teeth, actually. The nice thing about Sinatra is that you can relate this idea of variable selection with shapes to any kind of variable selection technique we do in genetics or any kind of basic area of statistics, which is actually really nice. And so I'll kind of go over that as well. And so Emily touched on it a little bit at the end, this idea of landmarks, right? And so the history of shape statistics and morphometric geometrics has typically been based on this idea of landmarks. So classically, we have some kind of three-dimensional shape. We place these landmarks that are either user-known or now they're more automated techniques to kind of do this. And then what we do is we kind of quantify or represent this entire shape based on just like the landmark points around it, right? And so biological morphometrics is a nice place to do this. These are with teeth from different primates where people in anthropology have gone in and they've kind of identified landmarks that they know to be to define the variation or the structure of this shape against the two groups. And what's nice about these landmarks that have allowed us to kind of do classical shape comparisons is now I can take these landmarks and I kind of can identify the distance between them, right? So instead of representing with this entire shape, I kind of just work with like the XY or the XYZ coordinates of these structures, and I can define distance functions to kind of identify or kind of understand how statistically significant the differences between these groups are, right? The thing that I don't like about landmarks, the things that's not useful for landmarks in my case, are two things. So first, as Emily had just mentioned, geometric morphometrics are often rely on these expert-defined landmarks, right? And so that kind of can bias me if I'm in a space where I don't really necessarily know where these landmarks should go, right? Or how many landmarks I should have, right? The second thing for me, which we're going to kind of talk about a lot and kind of refers back to topology, is a lot of these analyses require some kind of metric, right? So if I don't really know how large these landmarks should be, right? Size matters, right? To what metric I need to measure these landmarks to be, I could grossly misrepresent these shapes, and these can be kind of, my analyses and my inference can be biased by these like noises, right? And so one really nice thing about shape representations now is a lot of shapes can be represented as this idea of like meshes, right? Or collections of vertices, faces, and edges, okay? And I want you to keep this in mind because these triangularizations of these shapes are going to come to a big role in what Emily just talked about, this persistent homology transform, but as well as this Euler characteristic idea I'm going to talk about. As we sweep through the shape, these kind of mesh representations, we're going to take advantage of these kind of structures, okay? So the main objective of this talk is twofold. I'm really interested in this idea of representing shapes in some alternative representation where I can use a wide range of regression machine learning methods, right? I want to take my shape and I represent the information in the shape as like some vector, right? So that I can fit some kind of generalized linear model, or if I want to get fancy, I can do some neural network, right? But more importantly, I also want this transformation to be, injective, right? And I want to be able to compute and define probabilities in this transform space. Why I want the first is because as soon as I map my shape into some vectorized space, I want to then take the inference I do in that vectorized space and understand what that means in the context of my actual physical shape. Does that make sense? I want to be able to have this mapping back and forth. Okay. And so we're going to, Emily talked about three different really nice topological summaries. I'm going to talk about, I'm going to reference these three, but I'm really going to focus on the last two. Okay, so I'm really going to focus on this persistent homology transform idea with three-dimensional objects. And then I'm going to talk about why we created this Euler characteristic transform. Okay? So the nice thing about topology, kind of how Emily kind of mentioned, this is the idea of like this Picasso-type painting, is you can represent these like really complex structures in these like really nice lower dimensional spaces where we can kind of relate these lower dimensional spaces back to the difference of these like complex full data structures, right? And so just like before, for those who kind of stepped in and they weren't here for the primer, we talked about this idea of persistent homology, where persistent homology kind of tracks the evolution of these homology groups as a collection of sequential complexes, right? And so Emily mentioned this idea of as we start with these disparate connected component, we can kind of grow these balls around these individual dots here, and then track how these different homology features live and die as a representation of like barcodes, right? She also introduced this idea of the persistence diagram, and she also introduced this idea of filtrations, right? So I'm going to take a little alternative view to the type of filtration she showed you and stick more with this idea of like a two-dimensional image, how doing this filtration, we can kind of track the homology as like a birth and death time pair, right? So here we have this two-dimensional object. This is going to be really nicely when I project the three-dimensional objects to kind of get everyone in the right state of mind for 3D shapes. I'm going to start with my filtration here at the bottom, and notice how as I filter up, I'm going to count where a certain feature is born, and then I'm going to track where it dies via this square here. So here we have our self-filtration. We see our first feature. That's when it's born. I continue my filtration. This feature here dies, so we put a dot here, right? This kind of gets morphed into one as I kind of grow up. And I continue this filtration along via my sweeps, and I have this persistence diagram, okay? The nice cool thing about these persistence diagrams, as I mentioned this, I can kind of take these and I can do a myriad of different comparisons with them. So these are maze roots, where the interesting thing about different people who are interested in plants is the topology actually of your maze root can kind of tell you how well or how much yield a particular corn is going to produce, right? And so you can actually study in some like genomic selection type of way the topology of these roots and kind of compare and predict how much that particular maze is going to, how much yield that maze will produce. And so here we kind of do these sweeps. We get these nice persistence diagrams. And as I mentioned, you could take any of these kind of distance functions and kind of compare the differences between these groups, right? Then she introduced this idea of the persistence homology transform. And what's really interesting about this is it takes the idea of the two-dimensional sweep I just mentioned, I just demonstrated, and it now uses this in a three-dimensional object type of way, right? So instead of two-dimensional objects will not work with three-dimensional objects, right? She mentions this idea of doing this filtration with this K parameter, right? And then she showed this idea of the heel bone. Now, we're going to represent these heel bones as like triangular meshes. I mean, that's a question that kind of came up. And as I do this sweep across this thing with this filtration, I'm going to do that same kind of sweep and track the persistence diagram across that filtration, just in the same way I did that two-dimensional object. Does that make sense? So I do the sweep across. I take that persistence diagram. I save it, right? I do my second direction, another sweep. I take that persistence diagram and I save it. I do that for multiple directions, right? And so I can represent any shape as a collection of persistence diagrams. Is that a little more clear? Cool. So when she showed this kind of graph here, what was actually ended up happening was the fact that I compared to create this like two-dimensional principal component plot. This is based on a collection of diagrams, right? So the way that you understand the variance across these groups is I look at how the collection of diagrams differ as I do these sweeps in multiple directions for all these different 3D heel bones. Does that make sense? Now there are multiple issues with the PHT and the persistence diagrams in my opinion. One is the only way that you get anything out of this is if you actually know which genre these monkeys belong to, right? These are just points in space, and you have like kind of overlay knowledge on top of them, right? So I don't really know. If I know nothing about this, this plot means nothing to me. But more importantly, outside of that, common regression models, I can't necessarily take a persistence diagram or collection of persistence diagrams and do like linear regression with them, right? And it's actually a really simple concept. Outside the idea that there's no inner product structure with these groups. Another thing is like, if I have a birth and death time pair, what's an effect size mean for an ordered pair, right? Because in most of your in any kind of linear regression, I have like these vectors, right? But each vector has a single point in each cell, right? I don't have these ordered pairs representing like birth and death times, right? And so the interpretation of effect size is really hard when I'm working with like persistence diagrams, right? I can't necessarily. That's why the persistence landscape and all those other things try to vectorize these things, right? So what we came up with as an alternative, knowing these features, is that I kind of wanted something that's square integrable, right? I want to do something like the persistence landscape, but not only do I want to be able to relate my topological summary statistic to the shape, I also want I also want to be able to reconstruct that shape from my topological summary statistic, right? And so the Euler characteristic gives us the way to do that, and I'll explain how this works, okay? So the Euler characteristic is very simple to compute. I don't have this crazy infimum thing or anything like that. I'm just going to simply count the vertices, edges, and faces of my shape, and then use a simple formula here to understand what the Euler characteristic is of that given shape. Okay? Now, here's a demonstration of how the Euler characteristic curve works. So in the same kind of two-dimensional representation like I showed for the persistence diagram, I can also do something for the Euler characteristic, right? I can count how the Euler characteristic, or track how the Euler characteristic changes according to some filtration. Okay? So here's this hand. This is what I'm going to end up getting as my Euler characteristic. It's very easy. I start with the filtration. I can just sweep across. And as I sweep across, I can count as the Euler characteristic of my hand at each spot of my filtration changes over that sweep. Okay? So now what I end up getting, if I concatenate all curves, let's say I take that shape and I rotate my hand in many different directions, and I count the Euler characteristic all across these different directions, and I concatenate them, now I have just like one really long vector, right, representing the Euler characteristic of my hand in many different directions, right? And so my end result, after doing an analysis with the 2D shape, or even what I'm going to show you with 3D shapes, is just a matrix where each row is a concatenated EC curve for a shape in our data set.
Can you just go back quickly to the definition of the EC and explain why it makes sense that it's whatever minus whatever plus whatever? Like, I understand it's vertices, edges, and faces, but why does it make sense to look at something that's vertices minus edges plus faces?
Because of the mesh representation of our data. Because you have this nice triangular representation of our data. Now that when I do these, now everything, like these simplicial complexes idea, everything is like this triangulation type of measure. And so there's theory, which I did not come up with, of that these representations capture enough sufficient information like the persistence diagram does. There are limitations to this. So someone here asked earlier, how do I know, are there any kind of, am I perturbed, what are my weaknesses in this step? So there are obviously a few steps. Obviously in the Euler characteristic, more so than the persistence diagram, if my steps are not fine enough, or my steps along this filtration are not right enough, right, I could easily miss information, right? So there's this trade-off here of capturing just enough, right? The persistence diagram and persistence homology captures everything. The Euler characteristic is a nice like approximation to this idea. We capture enough information, maybe not everything, but I just want to capture enough to be able to do these kind of analyses that I want to do. What we want to do is we want to take advantage of this nice vectorization of things. And then we get this added bonus of being able, since I'm tracking at each step where the Euler characteristic is at each step, I can then map these things back onto these vertices at the end of my method. And so there's a give and take here with what is theoretically advantageous as well as like what is from an implementation perspective more advantageous. And so just like I was saying before, the Euler characteristic transform kind of gives us this collection of curves, okay? An EC curve has a nice simple inner product structure that we can take advantage of, right? So I can take my EC curves across all my groups. Let's say I have 10 hands. Each hand has a non-EC curve. Let's say those hands also have some like covariate or phenotype according to them. I can do linear regression really simply, right? Because I just have a nice design matrix and a Y output, and I can do anything you do in Stat 101, right? But I also do some really cool, complex, non-parametric regression methodology if I need to, right?
So,
yeah.
I guess that this would require that you somehow orient your various shapes in the same way.
Right, and so a big part of this is that you need to align your shapes correctly, right? Because the sweeps in this direction need to be such that they're comparable to all of the shapes in your data set. So there's a big case that I'm going to skip here. I'll mention it when I get to the teeth. But aligning shapes means a lot, and that's actually a whole other subset of image-based analyses, is how do you properly align shapes accordingly. We do this really nice algorithm based on this like unit sphere type of idea that aligns shapes pretty accordingly, but you're right, your inference can get really messed up if things are not aligned. So pre-processing here is a really big deal in all the examples I'm going to show you. So my application of radiogenomics is actually going to springboard the real part of this talk to me. And so here I have MRI scans of primary glioblastoma tumors from 4 different patients from the cancer, the TCIA, the Cancer Imaging Archive. These patients, the reason why we use this subset of patients is these patients also have matched genomic and clinical data along with them in the TCGA. So each, we get a lot of DICOM images that look like this. We do a nice segmentation, and then we make sure that we filter and pre-process all the DICOM images here, and then we segment just the tumors here because I just want to do analysis with the tumor segments themselves, okay? I'll show you a really pretty picture after this of how this has gotten a lot better since I first did this. And so after we have these nice segmentations, you have these slices of segmentations for each person, right? Then what we do is we do these sweeps with our Euler characteristic across different directions. So here are different directions here. Here's what our Euler characteristic curve looks like across the different directions. We concatenate them for each person, and then we're gonna do this prediction of disease-free survival and overall survival for each person. Okay, and we're gonna compare how well the topological summary statistics do as compared to like gene expression, or if I take in geometric measurements of this tumor or morphometric measurements of this tumor, and see how much information the topology is capturing versus these other notedly noisier summaries. So what we do in this paper is we use this idea of nonlinear regression, and I don't know how familiar people are with nonlinear regression. Nonlinear regression is much nicer than linear regression because instead of this additive relationship, right? So in linear regression, as you kind of know, right, you kind of have this like additive relationship between all of your covariates and the effects of all your covariates, right? Nonlinear regression just assumes that we don't have this nice structure, or this actually restricted structure, that my function that relates any of my covariates to my y can be anything, right? It can be some nonlinear type of, and this is a lot more flexible, right? So in this paper that I'm talking about now, and for the rest of the talk, we're going to focus on this idea of Gaussian processes, right? So I'm from Duke, and I'm a Bayesian by training, I guess. This f of x here, we're going to assume comes from, is we're going to put a prior on the function space where this f function lives, right? So Gaussian process, or a GP, is really easy to kind of understand. We have this like mean function of where we think this f of x kind of lives. We have this nice covariance function, or kernel function is probably what people kind of know this as, this kind of similarity measure between my given sample and all other samples in my data set, okay? So if this was gene expression, this K covariance matrix would say how similar is the gene expression of patient 1 to patient 2, right? In the cases of my MRI scans, what we're saying is how similar is the topology of the MRI from patient 1 than the MRI scans from patient 2, right? And we can do this in like a nonlinear fashion, where typically we set this to be like what we call an exponential kernel, okay? And so, like I just mentioned, we're going to compare this Euler characteristic idea to 3 key types of tumor characteristics: mRNA gene expression, tumor morphometry, and then tumor volume and geometrics. And we're just going to do this to predict disease-free survival and overall survival. To perform our prediction, we're just going to do this 80-20 training testing split. And we're going to do this like 100 different times. And it's going to use root mean squared error for prediction. Now, before you jump on me and ask me all these other questions about the difference between the biology and all this other stuff, I just want to kind of mention that we're going to use this as a springboard because it's really motivated me to kind of think about things moving forward. But here are the results from the paper, which are actually kind of quite interesting. We use the idea of a smooth Euler characteristic across all these different Gaussian process, different models, and all these different machine learning models, where we just changed the features that went into them. And each non-parametric method was actually able to learn the patterns of the topology easier than the patterns of the gene expression, morphometrics, and volume, right? For both disease-free survival, overall survival, not as much of a clear difference, but disease-free survival. I like prediction-based studies. I'm really more interested in this idea of like association mapping though. Like prediction's cool, but I don't really know what you do with this other than publish it, right? But what it kind of motivated me from a scientific perspective is this idea of, is there kind of any connection between shape variation and maybe biological phenomena, right? So this, what we call disease-free survival was the point that a person was operated on immediately after they got diagnosed to the time that the tumor came back, right? So it had some kind of relapse type of event. And so I got this kind of question of, is there kind of a way to relate this kind of tumor shape to this? As a side note, I also want to kind of point out how much better our images have gotten. This is the same tumor I showed before, but now we run each MRI through a convolutional neural network. And so now we can actually, from a background place from UPenn. And so now actually what we can do is we can actually construct meshes of tumors that look like I like gone in your head and like pulled out an actual like mesh of your brain. So now we're running all of our methods on like these type of analyses, rather these like 2D slices. So we've gotten a little bit more sophisticated as a group. But my bigger question, and what I've been writing grants on, is this idea of the can you use shape variations to explain genotypic or phenotypic variation, basically? And how do we build methods to do that? So that's where Sinatra comes in. Sinatra is this idea of using sub-image analysis for topological summary statistics. And I'll kind of explain like what that is. It's just a pipeline where I think you can kind of switch out all these different ideas. And we're not going to start with tumors because tumors are hard. So we're going to start with like teeth because I understand that a little bit better. So Sinatra has a nice pipeline involved, okay? So the pipeline is really simple. You start with any kind of shapes where you know these shapes are defined as classes. So shape 1 comes from one class, shape 2 comes from another class. These are teeth from primates. So this top tooth eats bugs. This top tooth eats plants. Bottom tooth eats plants. So these are different species according to evolution. What we do in our process of our pipeline is we take each mesh, we do these filtrations where we kind of we learn or we encode the Euler characteristic across each tooth, right? So we get these vectors or these curves that represent the variation of these groups. And then what we do is we do this nonlinear variable selection, which I'll talk about, which hopefully you guys think is also pretty cool as an inference and algorithm. And we highlight the areas of the curves that best describe the variation between these two groups. And then what we do with this reconstruction algorithm is we project onto the actual shape the areas that these curves actually came from, right? So then at the end, you get these highlighted regions of the spaces that kind of define the best describe the variation between two groups. Seems simple enough, I think. Cool. So like I said before, Sinatra's a pipeline, and why I like this is because a lot of statisticians say, like, why don't you use this method or not this method? So I'm really combative that way. So the idea is like, well, if you don't like any of these steps, like take out that step and then put your favorite pipe in, right? So to me, Sinatra has these 4 basic ideas. You represent some shape as like you summarize some shape based on their topology or geometry. You use some statistical model to kind of classify the shapes based on these summary statistics. Then you derive some like evidence of association for those topological or geometric features, okay? You can use p-values. P-values are not hot right now, so you probably want to use something else. And then you project these association measures back onto the original shapes, right? So I've kind of gone through the first already. We're going to use this Euler characteristic idea, okay? I also kind of already went through the second, right? I kind of mentioned this idea of like Gaussian processes and that kind of thing. So we're going to use this GP model to classify our shapes, right? Non-parametric methods are really good at, as you know, for neural networks and classification, they're really good at discerning patterns between two disparate groups. And then now we need to do these next 2 steps, okay? So my lab spends a lot of time thinking about this 3rd step from various different angles. We're really into this idea of interpretable machine learning, which I think you've heard a lot about after I looked on your website. But my group spends a lot of time trying to figure out how to take nonlinear methods and give them the same natural properties that you get from linear models, right? So linear models are really nice because they're really simple, right? You get this, you learn these things in Stat 101. You have hypothesis testing in this regime. Everything's really well thought out. But linear methods are missing this like nonlinear piece, right? And so my lab really thinks about how a lot of times in in a lot of like complex architectures of different traits, you don't just have this additive relationship of like gene A plus gene B gives you phenotype, right? You kind of have these also like these nonlinear functions that come involved where this gene A plus gene B, but also you could also have this effect of gene A times gene B. And so the reason why nonlinear methods do a really good job of prediction is because they kind of see the whole pie, right? Where linear models are really restricted, you don't see this whole thing, right? Now the interesting thing though is you have interpretability here, but you don't necessarily have interpretability here, right? Which is why all these people are trying to think about these kind of questions now. Another way to think about this is like this kernel trick issue, right? So I had this nice like Gaussian process machine that I kind of showed. The interesting thing is that when I do linear regression, right, I have this nice n by p model. Let's say that n is the number of samples you have, p could be the number of genetic features, genomic features that you have. People use this thing called the kernel trick where I take each x, I plug them into that kernel covariance function I showed you, like the Gaussian kernel type of idea. I get this nice similarity measure. I work now in this nice n-dimensional function space where here I do really good predictions because I see that whole pie I was talking about. And I do really nice predictions, but if you're any of us in this room probably, what you actually really want to know is not how to do good predictions, but how what each variable means in the context of that prediction, right? And so what you actually want to do is you kind of want to work here, but then go back to what that means in your actual input space, right? Unfortunately, what the big challenge is that there's this x here where this kind of relationship is lost, right? So the natural thing of like variable selection with nonlinear methods is lost on people. And so what I've really worked on is this idea of breaking this x and kind of understanding how to come over this problem, right? So here's one solution to doing this. I know you've heard a bunch probably at this point, but let's just think of like a different way. And if you don't want to do it, like I said, take the pipe out and put your favorite thing in, right? So one way to kind of think about this is that in linear models, you have this beta term, right? And in this beta term, you do all your like hypothesis testing with, right? You do t-tests, you do OLS with this, and then with this you get derive p-values, and with your p-values you do like multiple correction for hypothesis testing, and then you use those p-values to run off and do inference and whatever else, right? Nonlinear methods, you don't have that, right? You could just have this f term here. And so this idea of how do you relate what's going on in f to some idea of like a beta, right? And so we introduced this idea in this JASA paper, in this Annals of Applied Stats paper called the effect size analog, right? So in linear regression, in linear models 101, like I teach now, you have this idea of this projection onto the column space of x, which gives me this beta hat. This beta hat is used as like a weight for predictions. It's also used for hypothesis testing and inference, right? This projection is typically like a generalized inverse, or you use your generalized least squares, right? And I use this beta hat for everything. Interestingly, you can kind of do the same thing with nonlinear methods. I can have this, I have my nonlinear model here. Again, I don't have this beta term. But a cool thing for the effect size analog is that if you kind of think about it, you can project instead of this nice y, the nice smooth nonlinear function onto the column space of x, and get this idea of like an effect size analog of what happens after you kind of fit this nonlinear model, right? You can, if you want, use the same kind of standard projection, and you can use these beta tildes for the same kind of prediction-based or inference-based things that you would do with these beta hats, right? And I'll show you how to do the inference. The inference is a little harder here because we don't have nice theory about like statistics or anything like that that works with this beta tilde. But we do have this idea, this evidence measure called what I'm going to call rate. And I'll explain to you how rate works. Okay? Cool. Again, I said I'm Bayesian, so we would fit this with like MCMC or something. But the cool thing is that even in an MCMC type regression type method, every time I get a new draw of the posterior for my f function, I can also empirically calculate what that would mean in the context of beta tilde. Right? So I can get a nice idea of uncertainty that I'm getting from x and my beta tildes, right? As I get uncertainty in f, I also get uncertainty in beta tilde. Okay? So now I have these beta tildes, how do I actually rank them, or how do I figure out which beta tilde is most important? Because I don't necessarily get a p-value with these beta tildes, and I don't get a nice, what we call inclusion probabilities in Bayesian statistics. So what did I think about this? I realize Emily and I both like sports a lot. I think about this also in the context of sports. So one way to do variable selection is kind of in the context of the rest of the variables that you also measured in your data set. How I figure out who's significant should maybe be determined by who else I've also measured, right? And so let's take a team, again, any team. And let's say that this team has some distribution involved with it, right? I fit my data. I have a distribution of all the effects that this team represents, right, as a group. And let's say I take this random guy, I don't know, number 30 here, and let's say I put him on injury reserve, okay? Then I'm going to say how much information or how many games do I now lose with number 30 on the bench, right? Probably not that many, right? Now let's say I take another random guy and he goes and shoots Space Jam or he's going to play baseball or something, right? Then how I compare how important this human is compared to number 30 is I just say, well, how much information did I actually lose about my group with number 30 not around versus this guy who wants to play baseball not around, right? And I could do this iteratively for each person in my data set, and the members of the team that caused me to lose the most information are probably the best players on my squad, right? And so we can compute this actually really efficiently using this idea of KL divergence, okay? So what I do is I compute for each thing in my data set, for each variable or variant in my data set, I say, what's the difference of this conditional distribution with this player being set to 0, meaning that this player's effect being set to 0, or the marginal, meaning I have marginalized this player's information within the context of my group, right? If those two distributions are the same, or in other words, if the KL is 0, right, then this variant is probably not that important, right? If I look the same with you around versus with you not around, then you're probably not a really big player, right? Or in other words, this is only 0 if and only if these two distributions are equal, okay? Now, one way to think about this is that the KL divergence goes from 0 to infinity. So you can imagine, like if Jordan and Pippen were also both missing, then like they might equal infinity, right? So the way to kind of scale this to kind of figure out where an inference measure might be or a threshold significance measure might be is I can scale this based on everyone's contribution. So that's why we get this idea of relative centrality, right? How central are you relative to everybody else in the data, okay? The nice thing about this is it gives me a nice null distribution, and I'll talk about this a little bit more. A significant rate measure is anything in theory over 1 over p, right? Meaning, in the context where I have all scrubs or all bench players, right? Teams where like no one stands out from anyone else, everyone's going to be relatively equal, right? So in the perfect scenario where I have the replicate of everybody being exactly the same, I'm going to have this nice 1 over p idea where everyone's going to be, the relative centrality of each person is going to be across all p players, right? So anyone who's more significant than that is going to be far above that line, right? Does that make sense? Anytime I have a star, their rate is going to be above that. Yeah.
Does that rely on an assumption that the performance of each player is independent of every other player? What if two players?
Right, so it's a really good question. It relies on the assumption, so the null hypothesis for rate, and I'll show this in like multiple cases, is that every player is performing about the same. It's just that some players are more important than others. Does that make sense? That's the alternative. Right, so what you're saying is like, what if like I'm a star, but like I'm masked by Jordan, so when Jordan goes away, I become like...
Or what if two are always playing super well together?
Right. So that's a really good question. So the cool thing is about this measure is you can actually investigate that. You can take 2 players out iteratively at a time. Right? You can condition on taking out as many people together out as a time as you want, right? We've investigated this a few times. Very rarely do you see this masking effect. In social sciences, you actually can. When you actually, so going off topic, there's an interesting idea that for some social science studies, certain variables are masked to cover like other like ethnic type variables or other things. So like a certain question could actually mask the importance of the other groups. And so we actually could see sometimes is that when I do remove something, the rest of the importance of everything kind of like rises to the top. I'll show a simulation on the next slide, but very rarely do you actually see this kind of like masking idea. This idea where I'm only important given that this other player is important. The way that rate is calculated, it also takes into account this covariance structure. So you can be really important if like I'm not important alone, but I also interact with everybody together. So let's say like I'm the glue that holds the team together. I still will get a large measure because I also take into account your covariance with the other variables in my data set. So we've kind of tried to guard against just having this like marginal additive relationship, that you should be important in context of like what you're actually doing for the information as a whole. So here's a nice introductory feature of this. So quick simulation, I'm going to just simulate data with like 2,000 samples and 25 features. These last three are going to be like my Jordan, Pippen, Rodman, right? I'm going to have them all be a certain amount of effects, but I just want to show how rate performs in selecting these three measures and how this idea of this null hypothesis works, so you can see it before I go to shapes. So we run this data, we compute these rate measures for each variable. What you see is, again, you see Jordan, Pippen, Rodman here as a group. These three also interact with each other, so there's this interaction effect, so they're even more important than they probably would be, just marginally, right? Then what I kind of want to show you in terms of like what I mean by like this null case is, let's say I take Jordan and he plays baseball. What's really nice about rate is this idea that it's kind of intuitive in the fact that if I lose a star player, the importance of all other players should also rise, right? So if you lose a really key member of your team that like really was helping you carry this massive load, it's now up to the rest of everybody else to step up and also now take on more responsibility, right? So let's say I take out Jordan from the team, you actually see this effect happening, right? Still, Pittman and Rodman are the most important players, but the effects of all my other bench players has gone up, right? I need them now to step up and help carry this load, right? And we could do this all the way to the fact that I have no star players and I'm just left with a bunch of bench players, okay? As a sanity check, what you can see is that if I just took out number 30 or number 29 or some random players in each case, the effects of everybody is still the same, right? I still would dominantly choose these next three. The cool thing also is I were to simulate data where I just had everyone have equal effect, you kind of see this nice like null type scenario with some noise driven in. Cool, so what I want to do is I want to take this rate measure and this Gaussian process idea and use it for shapes. Now this reconstruction idea is really interesting. It's actually really intuitive. It's just a bookkeeping problem, right? I just keep, as I filter across, I bookkeep where each feature came, lived, or died as I kind of swept across my shape, right? And now I have these association measures attached to them. So when I project back onto the shape, I just take the rate measures of when they were born or died, or when they were seen as significant, and I project them back onto like the actual shape, okay? So this is kind of how it works. The goal of Sinatra, obviously, is to map selected features back onto the original shape. There's this cool idea from Kate Turner, Justin Curry, and Cheyenne Mukherjee, where they showed that the directions near each, if I take directions as my sweeps across the shape, directions within a cone of each other kind of tell you relevant information or kind of share relevant information. And so you can actually utilize that to kind of create this nice reconstruction algorithm I'm going to show you. So what we do is we use the following steps. We pick a cone set of directions. For each direction, I find all vertices that correspond to topological features selected by the GP. So I run my GP. I find each significant feature within that cone. And then I repeat this procedure for all cones. And then I take the union of all mapped vertices, right? So I only want to take the vertices that I saw evidence for in multiple directions. Those are the vertices that I feel most confident about, okay? And so this is easily how it works. I take my cone of directions. There are planes that come across them, and I just take the union of those planes to find those edges, those vertices that actually matter, right? I can do the same thing in any direction, and I take those. And at the end of it, I have this nice reconstructed based shape. So let me show you like really quickly for the last like 10 or so minutes, this nice simulation study using like perturbed spheres. One thing that I took for granted with this question or with this problem is how to do simulations. So in genetics, it's really simple. You just take real genotypes and you create synthetic phenotypes based on selecting like subsets of SNPs you think will be causal for the phenotype, whatever the case might be. With shapes, there's not this really nice variable selection framework, right? There are GANs and these there are these GANs that I can use where I can simulate like realistic shapes, but I can't control the features that are like causal, right? So we had a hard time in thinking about how to do this, and so I think we kind of came with a rigorous way to kind of think about simulation studies with three-dimensional objects. So a really nice proof of concept. I'm going to take 100 spheres, just blobs. I'm going to select a set of shared regions between these two groups, right? So I have 100 spheres. I split them into 50-50 splits. I'm going to take each sphere and I'm going to have a shared region marked by some cusp. So you see this nice cusp here. These cusps are going to appear in similar locations across all spheres, regardless of groups. And then I'm going to take these nice class-specific causal regions and I'm going to mark them by indentations. So I'm going to indent in these nice causal group-specific regions these indentations. So these indentations happen for this group. These indentations happen for that group. Okay. What I'm going to do is I'm going to assess the power of Sinatra via these like raw curves, which is true positives and false positives. Basically, how well is Sinatra able to find indices here and here versus like the false positive regions, right? What I'm going to do to make this analogous to what we do in statistics is I'm going to change the number of shared and causal regions between the shapes. So the more I make these shapes look different, the more causal regions I add and the more shared regions I had, it should get harder for Sinatra to discern between the two groups. Does that make sense? And that's actually, the intuition is actually what you see. So we have three scenarios where here's the easy scenario, which is just two cusps in one shared region. Then we increase it to four cusps and three regions, and then in something like seven shared regions, and then 5 causal areas. And what you see, similar to statistics, is in regular statistical settings, is when the number of shared regions is small, but the number of causal regions is also small. Sinatra does a good job of identifying this actual region here. This is actually kind of analogous to like, I always use genetics as an example. For polygenic traits, when you have many SNPs affecting a phenotype of each having small effect, it's hard to identify those SNPs of which ones are causal or not. Versus in more sparse type architectures, when you have a few SNPs with very large effects, it's easy to identify them, right? So the idea is that the variation explained by the causal regions actually means a lot in identifying which groups are identifying them. The nice thing is that this idea of Sinatra's, of rate's null hypothesis that I was explaining before, of rate assumes that everyone's contributing equally, and then its alternative hypothesis is that some regions are just contributing more. This also holds really nicely in this null hypothesis for two scenarios for shapes. In the case where I have two shapes that are totally similar minus some like minor perturbations with noise, every vertex here is effectively explaining the same amount of thing, right? There's no difference. So every vertex is basically equal weight, right? So you see so what Sinatra says is that it can't identify the difference between two groups, right? So when everybody's the same, every vertex is also the same, meaning that there is no signal. Sinatra's going to say there's just like everything looks significant in this case, right? Same thing when two shapes are totally different. If two shapes are totally different, every vertex is explaining the variation equally between two groups. Sinatra is also going to say, I can't understand what's really going on. And so there's this nice middle sweet spot of the shapes have to also be slightly comparable in some way, right? And so we kind of learned a lot about this here. We did a second simulation scenario where we did a little bit more sophisticated setting where I took CT scans of real teeth from primates. The classes are defined by creating like causal and shared regions the same way I did with the spheres. But this was done by modifying regions of interest that we know from expert-derived landmarks, right? So there are these, and then we're going to assess Sinatra the same kind of way. I take a tooth where I know that this tooth has certain landmarks, and I'm going to perturb these different landmarks in different ways to create my two groups. Okay, so I select 3 landmarks here for class 1. I select a different set of 3 for class 2. I perturb them in different ways to create my class 1 and class 2. Like I exaggerate them via this characterization algorithm. So this creates my shape class 1, my shape class 2. I run Sinatra on it and identify how able it is to identify these regions here and these regions here as my true positives and false positives. Simple enough. Same thing, when I increase the number of landmarks to perturb, meaning I make the shapes harder and harder to recognize across the groups, once again, you see this drop in Sinatra's ability to kind of find them, the same kind of intuition that you would have for regular statistics, right? As the variation is split among many more players or many more variants, it's hard to determine who's the most important. And the last thing we did is we did this CT scan stuff with real data. So here I have 59 molars from 2 different suborders of primates. We're going to focus on this Tarsius, and the reason why we want to do this is because we have some like ground truth here. So again, the hard part about this method is trying to find things to compare that had like ground truth, since we were like kind of the first people that kind of thinking about this. The ground truth here is that the Tarsius have retained this nice feature in their teeth that allowed them to choose certain foods that other primates have not via evolution, right? And so the first goal is if Sinatra can identify or recover this specific feature on the lower molar, and the second is if Sinatra is identifying this, is it easier to do this and relate this back to like the variation across molars that you see according to divergence of time in evolution for these monkeys. And so here's a Tarsius at the bottom. Here's this kind of phylogenetic relationship between these groups. Here we have the different teeth, and we see some, this nice red region here is the known feature that we're trying to identify. Our hypothesis about this via like our simulations, in the case that we know where the null setting would happen, is that these two teeth are really similar. So I think we thought it would be harder for Sinatra to identify this region when compared to these Samiri here versus the other 2 primates, which are a little bit more different. And so identifying this feature might be a little easier. And that's actually exactly what you see. Here you see there's a little bit more prominent red regions here for this cusp than you do for that Samiri notation. What we actually did, and then we went through and we did some like really cool hypothesis testing procedures where we did some like null experiments where we try to identify via like p-values how likely that we found this region by chance. But I won't get into that here. And so ongoing work in the lab, and kind of why I was excited to come here and talk about this, is we've been exploring Sinatra with multiple things. And so one thing that's actually ongoing right now that I hope we publish or we hopefully submit in the next couple weeks is this idea of exploring Sinatra with deep learning methods. And so Sinatra's like this vectorized, the Euler characteristic gives you this vectorized topological summary statistic. We've kind of shown is using like deep GPs, the classification of really complex shapes gets a lot easier than just using this regular Gaussian process. And so neural networks or even any kind of deep learning algorithm actually has been really nice. The cool thing about rate is you can extend rate to these nice deep learning type of algorithms very simply by just taking this probabilistic view on them. And really more importantly here is I kind of want to move Sinatra to this more biological driven shapes in medicine and radiomics. And so we've been trying to think about how to use Sinatra or derive summary statistics for MRIs and things like that. What's really hard about tumors is that these things are really highly dissimilar. And so topology alone, I don't think encodes the variation that we want them to. And so we've been thinking about this idea of, when you think about like 3D microscopy images, MRIs, things where we have like texture involved, how do you also augment the topological variation and the geometric variation that we're getting and augment that with more texture-based features that people do with these type of images now, right? So how do you kind of mesh these two and still these like variable selection-based ideas? And so that's one thing that we've really been thinking about a lot. And I think this will like only open up the use case for Sinatra if we can try to use these things in these type of domains. And so any kind of thoughts people have about that stuff, or if anybody wants to work on that stuff or write down 95, it'd be great to like think about that kind of stuff. So with that, I kind of want to thank everybody for hanging out. This Sinatra work was actually led by an undergrad and a master's student at Brown. He's now a PhD student at Princeton, and then a PhD student at Duke. I'm funded by people and other collaborations. And thank you guys so much for listening and hanging out. Appreciate it.